Okay, so we're going to talk about the two methods of DNA sequencing. This is the first one, the original Sanger method. This is actually what I used to do when I was doing my master's. So it was still used, let's see, when did I finish? 99? 1999, they were probably just getting started, or um, the newer method was just starting to take hold, and some places could afford to have the machines to do it, but it was fairly new at the time. So um, this was the original method for DNA sequencing that was used. Um, Basically, what they would do is they would take the DNA and they would um, use a primer or a little sequence that would kind of get replication started. And they cause DNA replication to happen in 4 different tubes. So within each of the 4 tubes, they would put a special molecule along with all 4 regular nucleotides. They would put special nucleotides that kind of stopped replication at different places. So you'd end up with all different possible lengths of the DNA that ended with each possible letter. You're not going to have to be able to explain exactly how it's done. This is just kind of giving you an idea. So because it cuts the DNA in all the different possible lengths that end with, say, an A, when they run that tube of stuff in a special gel, then the little sections are going to be able to move along the gel depending on how long they are. Okay, so the gel runs this way, going down. So the shortest pieces, or the ones at the first, end up going all the way to the bottom because they're really fast when they run through the gel. They're charged and they move towards the other charge. And so the 5' end, or the start of the DNA section that's being sequenced, is at the bottom, and the gel is read from the bottom to the top. Okay, so if you were reading from the bottom to the top, then you do so by going from the shortest. Okay, so the A is the shortest, it's going to be the first. Why am I using that? Silly, I'll just use the pen. Okay, so A, and then you move over, and the next shortest one is here. Okay, so the next letter is a C, and then the next one is over here, which is under in the T reaction, so it ended in a T. And the next one would be a G, and then a C, and then a C, and then an A, and then a G. Okay, you get the idea. So you read it from the bottom to the top. That's a G. Oop. And then C, C, T. Okay. If I were to ask you to write it from 5', which is the first of it, to the 3' end, then you'd have to tell me from the bottom to the top. That's a 5. Looks like a 3, but it's supposed to be a 5. Okay, so it starts with A, then C, T, G, C, C, A, G, T, G, G, C, C, T. And that's it. Okay, so you may have to do that on the exam, but it is definitely going to be on that little mini lab. So you read it from the bottom to the top, and that's the 5' end to the 3'. Now, some things about this particular sequencing method where it required that they use 4 reaction tubes as opposed to what you'll see in the next one. We had to use radioactive labeling, so we had to deal with radioactive substances, which is, if you can avoid that, that is healthier for you. But we did take special precautions, and we had to wear special equipment, and in some ways it was kind of scary working with it, but as long as you are using all the safety precautions, then you should be okay. Still, if there's a method that you can use that does not require radioactive isotopes, then that's better. The third thing was that humans had to read the sequence. Okay, so because humans are involved in the actual reading of it, there's a chance, maybe you make a mistake in some of those lines. But the second method is automated sequencing. So you basically prepare one reaction tube. You still have molecules that stop the reaction at every possible length, but this time the the um, the primers themselves are labeled with fluorescent labeling and fluorescence. We don't you don't have to worry health-wise with fluorescence. Okay, so they're fluorescent labeled instead of radioactive. Um, Computer is going to detect it. Okay, so the computer detects it, and then you get, this should make it bigger. You get a printout that shows all of the sequence at once. Okay, so the computer does the work for you. Let me see. I need this one. I think that, there we go. And this is what it looks like. Okay, so if you were to read it, oops, if you were to read it, it shows you the sequence right along there. Okay, from all the different peaks. So the computer does the work for automated sequencing. You just have to prepare the original tube, one reaction tube. And I think, oops, yep, that's it. That is the end. So that's the last lesson for grade 12 biology. We have a little mini lab to do, and then you'll be doing some review activities, playing some of the review games over again if you want to, or working on or getting a chance to look at the multiple choice booklets again. You won't have time to write things down from them, but it'll give you a chance to look at the answers. Give you some time to start your studying, make study tools, and yeah, just try and make sure that you're all ready. Don't forget that you can text me or send an email to my phone. Miss Power has the address and the phone number so that you can do that. Oh, I could just put them on here. Okay, so my phone, if you're gonna text me, is this number. And if you're gonna email it from your email, then it's that at, have to double check with Miss Power to make sure I'm doing this right. GudoMobile.com. I think that's what it was. Okay, so Good luck when you're studying. Take your time. Put stars by things if you do have questions. Try to help each other, and don't be scared to text me or email me, and I'll do my best to get back to you. Um, it's been great having you guys in class. I always brag to everyone how I'm the luckiest teacher because I always have the greatest groups. So thank you so much, and I hope I'll be in before everything's done, so that I can introduce you to Charlie, but also to see you again before we're officially all through of biology. Anyway, take care. Make sure you get some rest and eat, and change your scenery once in a while when you're studying for exams. And I will stop because I know I'm ranting and ranting, but it's the mother in me. Good luck. I'll see you soon.